name is Dr. Simon Thebault. I'm a neurologist and clinician scientist at the Montreal Neurological Institute. I'm a PI with the Ludmer Centre, and my research mainly focuses on neuroimmune conditions such as multiple sclerosis. I see patients for a quarter of my time, and the remaining time is spent doing research. I guess I've always been interested in the intersection of neurology and immunology, but it wasn't until I got into practice that this became a research interest. I already had a wet lab background from a master's in Oxford in the UK. So during my residency in Ottawa, I became exposed to patients with multiple sclerosis and was really struck by how encouraging a field this is. In the last 20 years or so, there's been a number of therapies that have revolutionized the care for patients with MS. And we have the opportunity now, new innovations, single-cell technologies, for instance, to really get a deeper, more granular profile of individual patients. My vision is that we develop and create and even implement a biomarker signature which, in addition to providing diagnostic information, can help predict which treatments a given patient might respond best to. So there are several reasons why I chose to come to the Neuro and to McGill. First, I think nowhere else has a better vision and history of translational research than the Montreal Neurological Institute. Secondly comes the resources like CBIG Biobank, the Ludmer Centre, a world-renowned imaging group, the institutional infrastructure at McGill. And thirdly, and to be honest, the most important reason is the quality of the people. I've got some great collaborators. I felt that I was a piece of a puzzle, and I could see exactly where I'd fit into this puzzle. Recently identified imaging signature of worse MS called a paramagnetic rim lesion. Around half of patients with PERLS have a worse prognosis, and we think that the reason for that is PERLS, a chronically inflamed lesion, and this smolders over time, leading to ongoing disability. We can actually identify a biomarker signature at the immune cell level associated with more PERLS within a given patient. If around the time of diagnosis, we identify this signal, that's maybe an opportunity to intervene and prevent disability. The virus that we study in the lab that's been associated with the onset of MS, Epstein-Barr virus, infects almost all of us, but it establishes, like other herpes viruses, long-term latency. In the case of EBV, that long-term latency is in the B cell. All patients with MS seem to have this temporal relationship between developing EBV and then subsequently developing MS. But most of us will get exposed to EBV and develop a normal immune response. But we know that people that develop EBV much later and who develop a more severe form of infectious mononucleosis seem to have an even higher risk of developing MS. And we've detected a signature related to more EBV reactivation that coincide with recent disease activity in MS. Can we identify the EBV-infected B cell in the brain to inform as to how this biomarker signature that may be linked to the driving of the disease itself? Key people that I work with include Dr. Adil Harood. He's a neurogenetics researcher here at the Neuro. I'm excited to be exploring the interaction between genes and the protein responses that we can see in the cerebrospinal fluid and in the blood, and how can we integrate genetics into some of the biomarker measurements to give us the best approximation of risk profiling. I work with Dr. Joanne Stratton and Dr. Stephanie Zandi. Dr. Stephanie Zandi works on post-mortem tissue. The idea would be to develop a signature that we think is associated with worse outcomes and then validate that that is actually mechanistically relevant in the multiple sclerosis lesion. I'm also working with Dr. Ciro Piccirillo down at the MUHC immunology group, and he has some models regarding T regulatory cells, as well as he's interested in our work on Epstein-Barr virus and how that might relate to other non-neuroinflammatory autoimmune diseases. Other key collaborators include Dr. David Rutko and Dr. Sridhar Narayanan. They're both MRI researchers. It's all about how can I associate the immune cell measures with some of the advanced imaging outcomes that they can detect. Our most widely used therapy is B-cell depleting therapies. Patients have continued on these for many years, and we're only just starting to learn about what effect that may have on the immune system. I've started working with Dr. Michael Seabag. He's a hematologist at MUHC, and we've started working on CAR T-based therapies. We actually take a patient's own immune cells and modify those cells and then give them back to the patient in a more precise and targeted way. Some of our drug treatments don't cross the blood-brain barrier at high enough concentrations, but perhaps these immune cell-based therapies cross over into the brain, into the lesions where lots of this inflammation is happening, and alter the immunology in a more durable fashion. joined the lab is an exceptional privilege because my lab members, they're not just employees, these are my trainees. Our fate is closely tied together. So I've got a research associate lab manager who has a PhD in molecular biology. I also have a postdoctoral research fellow who came over from the HIV field. She's applying her techniques to EBV. In addition, I have an MD PhD student who's working on the protein biomarker side, and he's a key member in terms of that intersection between understanding the clinical language and being able to link that back to patient-related outcome measures and imaging techniques. And finally, I have a shared lab member with Dr. Stephanie Zandi, who's focused on regulatory T cells. One of the key things that gets me up in the morning is regular patient contact to keep things real. There's one thing studying abstract patient samples, but many samples actually come from patients that visit our clinic and are my patients. That can provide a vision of the future direction to the patients, and it's a hopeful vision. And similarly, on the back end, we've already had the privilege of implementing some of the early development work back into the clinic. So there's an example of a new test. We didn't discover it, but we've locally validated it, and it's now starting to be used routinely. Another test that we focus on is called neurofilament light chain, and this is a blood test that we can use and serially check in patients over time. And this would be an additional tool to help with the disease activity monitoring, in addition to the MRI scans that we already do. And that's on the cusp of being put into routine clinical care. We work closely with the clinical research unit here at the Neuro, which is a fantastic team dedicated to clinical trials. So that provides an additional opportunity to study many of the immune cell and biomarker measurements that we're very interested in to help with that translational side to what we're doing. So really, the clinic is both the beginning and the end of all of the questions and the reason why we're doing what we're trying to do in the lab. It's a long and winding path to get through medical school and the subsequent training alone. To get through that and still have energy left and be interested in being a scientist as well is a rare thing. And providing opportunities to develop those interests is something we really strive to do in the lab. And working at McGill, there's no shortage of excellent medical students, resident trainees who we can involve in the research.